Today we're going to talk about synaptic transmission. And synaptic transmission is about how we get a message from one cell to another cell across that little space called a synapse. So let's go ahead and begin by orienting ourselves. I've drawn two cells up here, two neurons for example. And the first neuron we're going to refer to as the presynaptic cell. So we'll go ahead and label that presynaptic cell, and then we're going to have our synapse. Now a synapse consists of the end of one cell, the actual space, and a little bit of the beginning of the next cell. That's going to be our synapse. Over here, this is going to be our postsynaptic cell. And this postsynaptic cell could be a neuron, as I've drawn here, or it could be a muscle cell, for example. Now, let's go ahead and talk about what is actually going on here. A signal is going to move down the neuron, and a signal moves down the neuron by a process called an action potential. However, once that action potential, once that electrical signal gets to the end of this neuron, once it gets to the axon terminals, something different is going to happen. And that message, that signal is going to get transmitted across the synapse. And that's what we really care about today. So let's go ahead and focus in on this synapse. So I'm going to go ahead and enlarge this area. I'm going to go ahead and draw a synaptic, or not a synaptic term, I'm going to draw an axon terminal. That's going to be an axon terminal. So that could be this right here. That could be that. And this is going to be our presynaptic cell. Now, in an action potential, you're going to have various proteins. This is going to be our voltage-gated sodium transport protein. This is going to be a voltage-gated potassium transport protein. And this is going to be a sodium-potassium pump. And these proteins repeat themselves over and over and over down the membrane of the neuron. Now, once we get to the end of an axon terminal, these proteins are going to change a little bit. And we're going to get a different type of protein. And I'm going to draw one, but there could be tens to hundreds of these proteins all around the end of this axon terminal. And this particular protein is going to be called a voltage-gated calcium, Ca2+, TP, transport protein or channel protein. We'll call it a transport protein. And it's voltage gated. That means that a change in voltage is going to cause this protein to open up. And when it opens, because it's a calcium transport protein, and they tend to be very specific, calcium is going to pour into the cell. So we're going to go ahead and draw that. This is calcium pouring into the cell, Ca2+. And outside the cell, there is lots of calcium ions. And inside the cell, there are very few calcium ions. So once this protein becomes open, the calcium is going to want to move down its concentration gradient. So it's going to want to move from where there's a lot to where there's less of it. So it's going to move into the cell. And this voltage-gated calcium transport protein is only going to stay open for maybe a few thousandths of a second, and then it's going to go ahead and close again. But a lot's going to happen in that few thousandths of a second. Now also, inside this axon terminal are going to be some membrane-bound structures. And these membrane-bound structures, these are transport vesicles. And because these transport vesicles are found at the end of the axon terminal, we're going to give them a special name. We're going to call them synaptic vesicles. Now inside the synaptic vesicles are going to be some proteins. Lots and lots of proteins. And that's what these red dots represent, all those proteins. And the proteins are going to be acetylcholine. That's going to be the protein. Acetylcholine. The acetylcholine is also a small molecule, which is known as a ligand, and we'll get to that very soon. Now the calcium is actually going to come into the axon terminal, and it's going to trigger these synaptic vesicles to move to the surface of this axon terminal. And I'll actually put a transport vesicle or synaptic vesicle right there. And when the synaptic vesicle merges with the surface, with the cell membrane, it's going to dump its contents into the synapse. We call that exocytosis. We're going to exocytose all that acetylcholine into the synapse. Now I'm going to draw my postsynaptic cell. And my postsynaptic cell, here it is. That's going to be my postsynaptic cell. Now again, this could be a neuron or it could be a muscle cell. If this was a neuron, we'd actually be looking at the dendrite. If this is a muscle cell, then we'd be looking at the muscle. In this postsynaptic cell, we're going to have some additional proteins. And I'm going to go ahead and I think I'm going to use brown. See if that shows up. And there it is. And this is going to be a special transport protein. This transport protein, and again, there's tens to hundreds of these transport proteins around here. This transport protein is going to be referred to as a ligand-gated sodium transport protein. Now, a ligand is a small molecule. And the small molecule in this case is acetylcholine. And we call it a ligand-gated. So this transport protein, the opening of it is controlled by the binding of a ligand, or in this case, the binding of acetylcholine. So acetylcholine is going to diffuse across this synapse. Let's go ahead and label that. And it's going to bind to this ligand-gated sodium transport protein. And when it does, it's going to open it up temporarily. And when that happens, the sodium is going to pour into the postsynaptic cell. Now, just like action potential, there's a lot of sodium outside the cell because of the sodium-potassium pump. So there's lots of sodium outside the cell and very little inside the cell. So when you open this transport protein, the sodium is going to flow in. And it's going to continue to flow in until this ligand-gated sodium transport protein shuts. Now, within a few thousandths of a second, it should shut. And the reason it should shut is because we're going to break down that acetylcholine. What isn't shown here is an enzyme. In fact, there's a lot of this enzyme around here. And this enzyme is called acetylcholinesterase. And acetylcholinesterase breaks down acetylcholine. And when we break down the acetylcholine, this transport protein is going to shut and prevent more sodium from coming in here. However, let's backtrack a little bit. The acetylcholine diffuses across the synapse. It opens the ligand-gated sodium transport protein. Sodium rushes in. The voltage in this area changes. And that change in voltage is going to trigger other proteins to then respond. So let's go ahead and draw some other proteins. Around here are more proteins that we talked about during the action potential. A voltage-gated sodium transport protein, a voltage-gated potassium transport protein, a sodium potassium pump. And the same proteins are found down here, the same proteins are found here, and the same proteins are found here, and again, and again, and again. So when the voltage changes in this area, these transport proteins feel that change in voltage. That's why they're called voltage gated, because they respond to a change in voltage. And when they do that, they're going to open up, and that's going to create a change in voltage. And when the change in voltage occurs here, then the next set of transport proteins feel that change in voltage, and they begin to respond, and so on and so forth. And we, again, begin another action potential down the muscle cell membrane, or in this case, down the neuron. Now again, we've got an action potential going on here, and then when we get to the very end, that's when we get to synaptic transmission, the opening of the voltage-gated calcium transport protein, which then causes the synaptic vesicles to fuse with the cell membrane, exocytosing the acetylcholine. The acetylcholine binds to the ligand-gated sodium transport protein and allows the sodium to flow in, which changes the voltage of the postsynaptic cell. That process I just described is referred to as synaptic transmission. As soon as the voltage changes here, that initiates another action potential, and the signal, the action potential or electrical signal, continues down the postsynaptic cell. And that's going to be it for synaptic transmission.